Pharmacognosy, between past, present, and future. We use it in our daily life, in treatment and medicines, and even in food and drinks. It is the herbs. So, when did humans start using them, and how did he know about them? It all started in the Stone Ages. The primitive man started to know plants during his journey to find food. At first, he was eating plants only for feeding. Then, he began to notice that some plants have beneficial properties and used them to treat some diseases. He also found out that some plants have toxic effects, so he used them in his arrows for hunting. Human has developed and different civilizations have emerged. Among these civilizations was the Indian civilization. In the period between 3500 to 1500 before Christ, medicinal characters has been registered in two books, Rigveda and Atharvaveda, which created a system called Ayurveda. Ayurveda, which means science of life. It was a system of a traditional medicine. Although it was originated from South Asia, but it spreads all over the world, and its techniques are still used until now as yoga, massage, and herbal medicine. After that, Chinese civilization was founded. It contributed significantly to the plant-related sciences botany. In 2700 before Christ, Emperor Shennong has discovered the medical value for hundreds of the herbs. Also, he wrote the first book of herbs, Bin Tao Ding, in which 365 types of medicines had been written down. After that appeared recipes from the Golden Chamber, book by the scientist Qin Kai Yao. Hence, it was the foundation of herbal science by the Chinese. Also, in the Pharaonic era, specifically at 1550 before Christ, the Apis Papyrus was written, which contained 800 medical recipes and 700 medicines. Anthem's Papyrus, which was considered as a reference of how to conduct surgeries and prepare cosmetics, and contained the most commonly used herbs, such as senna, thyme, cumin, and caraway, and also Papyrus of Cones, which was one of the oldest papyrus addressing the women's health and childbirth. As for Greeks and Romans, they have greatly contributed to the development of knowledge by natural medicine. Among them was Hippocrates, who was called the father of medicine, who doctors repeat his famous oath as the first step to practice the medicine. Also, the Roman civilization has introduced a number of scientists, including the scientist Aristotle, who was a philosopher, and also he was known for his writings about the kingdom of animals. Then his successor Theophrastus wrote books about the plant kingdom. Later on, in the 1st century, Anno Domini physician described the medical properties of plants, some of which are still being used until now, such as belladonna and onion. In the 2nd century, Anno Domini Galenius described the methods of preparing drugs from plants and animals known after his name. The beginning of the 19th century was the actual beginning of pharmacognosy. On that century, scientists separated the specific active constituents from the plants. The term pharmacognosy appeared for the first time on that century. In 1803, French pharmacist Desarone was able to extract narcotine from the opium plant. In 1806, scientist Cerner extracted it from opium as analgesic. In 1811, in Vienna, Dr. Schmidt was the first one to use the scientific term pharmacognosy as a description of medicinal plants and their properties. Pharmakon, which means medicine, and the term gnosis, which refers to knowledge. We can define it as the science which specializes in the study of raw drug materials, and it's also linked to plant anatomy and chemistry. By the end of the 19th century and the beginning of the 20th century, pharmacognosy became a branch of applied science with theories and technology. In the 20th century, modern methods appeared to identify the constituents of chemicals and natural substances, resulting in reaching to accurate information about extracted natural materials. This includes the science of spectroscopy, what we use to know the number of atoms of the compound, the number of its molecules, and the type of bonds that make it up, which gives a complete picture of each compound. A colorimetry technique also appeared. to measure the concentration of colored matter in a solution using a device that measures the amount of light with a specific wavelength which had been absorbed by the solution of unknown concentration. In 1928, William Fleming extracted penicillin from penicillium mushroom, which made a huge revolution in microbiology and started with it a new world where death is not inevitable by bacterial infection, and the tragedy of tuberculosis and cholera had ended. Scientific achievements subsequently followed, and many substances of high medical value appeared, including reserpine, which was extracted from Rauwolfia root, and confirming its tranquilizing and hypotensive properties. Vinblastine and vincristine were also extracted from Catharanthus roseus, which were considered as a very effective anti-cancer drug. At the end of the 20th century, pharmacognosy became the foundation for various sciences, rather than the study of natural materials. For example, pharmacognosy began to play a role in the discovery of forensic evidence, and forensic botany has originated, where botanists were able to come up with information useful for solving mystery of the crime, such as the time and location of the crime after examining papers, seeds, and pollen. If they are found on the victim's body or in the crime scene, this technique is also used in drug trafficking cases, as it's possible to know the source of drugs through pollen grains that are attached to the shipment. Also, biotechnology was closely related to pharmacognosy, through which it was possible to obtain controlled agricultural crops in order to obtain the greatest medicinal benefit from plants. Through biotechnology, it was possible to produce plant strains that resist pests and strains that contain a higher concentration of the active substance. For example, the production of golden rice that contains 3 genes that allows the plant to produce compounds that are converted into vitamin A in the human body, and that was designed to fight the main cause of blindness in the world, which is the deficiency of vitamin A. Also, through biotechnology, the problem of malnutrition was addressed in Uganda, as genetic changes were made to bananas to contain vitamin A and iron, and this contributed to solving that problem. To make the world green, pharmacognosy contributed to obtain green or bioplastic, as it was made from plants such as sugarcane, wheat, corn, and potatoes. And this type is environmentally friendly, as there are some types of it that are organic biodegradable and disappear without leaving any harmful impact on the environment. Also, one of the greatest techniques applied today is tissue culture technology, which is based on the findings of the German botanist Heberland in 1902, that any plant cell can give a complete plant. Currently, this science has been applied widely, as any part of the plant, regardless of its size, is separated even if only one cell and is planted in the laboratory in certain conditions. So the spore grows into a whole plant, which is multiplied in a next step in order to obtain the largest amount of active substances in the shortest period of time. This science also allows for the desired genetic modifications to the strain of the plant being cultivated. Now we can say that the world is still waiting for a lot about plants. There are 1.5 million plants around the world, most of them not yet studied. And problems with the side effects of medications are increasing day to day, which forces the world to find a safe alternative. Perhaps more studies about plants, animals, and minerals that are still unknown is the world's hope for this safe alternative. Finally, we must remember that all the efforts of science are still considered to be an outer layer of nature, and there are many we haven't yet reached and know nothing about it.